The sphingolipids are another major component of cells. The sphingolipids you can see schematically on the screen. They contain a compound called sphingosine that we'll see how is synthesized in a minute, and that sphingosine is linked to a fatty acid shown below in green. Now, the attachment material point for the sphingolipids is the R that's shown on the right of this molecule. So sphingolipids are important, especially for nerve tissue and brain. So very, very prominent components. Sphingomyelin is one of the principal components of the myelin sheath of nerve cells. Sphingolipids are comprised of sphingosine, a fatty acid, and an R group, as I said. So these will all come together to make the amphiphilic substance that comprises the sphingolipids. Now, the synthesis of these compounds starts with very simple materials. It starts with the amino acid serine and palmitoyl-CoA. These two are joined together to make something like the sphingosine, as we shall see. So here's the amino acid serine, and to that we combine a palmitoyl-CoA. The product of that reaction is dehydrosphingosine, and you can see that we split off a CoA and a carbon dioxide in the process of that. Now, dehydrosphingosine can be converted to sphingosine. Now, sphingosine is not directly converted into the sphingolipids. Instead, dehydrosphingosine is, and it's dehydrosphingosine that is modified to become dihydrosphingonine. A mouthful of names here. Dihydrosphingonine is reduced in some reactions, as you can see here, and a CoA is attached to the molecule. The product of that reaction is something called a ceramide. Now, a ceramide is a building backbone for a sphingolipid, just like phosphatidic acid was a building backbone for a sphingolipid. The R that you can see on the ceramide at the bottom is the R that's in the upper portion of the figure on the screen. Now, the R group attachment point is where the differentiating components of the sphingolipid will exist. If the R is phosphocholine, or if the R is phosphoethanolamine, we create one of a class of compounds called sphingomyelin. So as you can see, sphingomyelin is not one compound, but a variety of compounds. These sphingomyelins comprise the lipid bilayer of the myelin sheath of nerve cells, so they're very, very important compounds. If R is a single sugar, then we've created what we call a cerebroside. And like its name would suggest, cerebrosides are found commonly in brain tissue. If the R is a complex sugar, then we create what's known as a ganglioside. This concludes the discussion of the metabolism of the sphingolipids and the glycerophospholipids. I hope that I've communicated to you that these compounds that are found in the lipid bilayer, like many compounds found in cells, have roots in very simple molecules but derive very complex functions.